Welcome to our second online classroom. In this lesson, you'll be learning more about meiosis. Before you begin, get ready your writing materials so that you can jot down important notes as well as answer some question posed. Feel free to pause the video whenever you need more time to digest the information, okay? Just like in any bio topic, you need to know the definition of certain terms. For meiosis, it is a form of nuclear division such that the daughter nuclei produced contains half the number of chromosomes as the parent nucleus. Meiosis have more stages than mitosis. To make things simpler, it is divided into two divisions, Meiosis I and Meiosis II. Meiosis can occur in both animals and plant cells. What you're looking at here is actually a photograph of meiosis occurring in onion cells. Pretty interesting, isn't it? Just like in mitosis, meiosis also consists of 4 stages, namely prophase, metaphase, anaphase, and telophase. But before the cell can undergo the 4 phases mentioned in the previous slide, it will have to first go through what we call the interphase. This is where the chromatin threads replicate, producing 2 identical sister chromatids attached at the centromere. This is, however, not visible under the microscope. Now, if you look at the top part of the cell, you will notice 2 tiny structures. These tiny structures are called centrioles, and they only divide in an animal cell. As mentioned earlier on, meiosis comprises of meiosis I and meiosis II. So for now, we will focus on meiosis I. The first phase is called Prophase I. This is where the thin chromatin threads condense, coil, and shorten to become chromosomes. There are actually 2 parts to Prophase I. The first part is called early Prophase I. This is where the homologous chromosomes pair along their whole length. This process is called synapsis. Now, what are homologous chromosomes? They are actually chromosomes that have the same shape, same genes, and same length. However, one of the chromosomes comes from the male parent, less confusing, while the other we have from the female parent, the chromosomes. So you can see the red strand represents chromosomes coming from the female parent, while the green strand represents chromosome coming from the male parent. This color coding will be the same throughout this lesson, so do take note. Now let's focus on the diagram again. If you look at the top part of the cell, you will see these tiny structures called esters. Esters appear around the centrioles, which move apart to opposite poles of the cell. Now we'll move on to late Prophase I. As you can see, the chromosomes were shortened further. Each chromosome is now seen to consist of 2 chromatids attached at the centromere. Chromatids of homologous chromosomes may cross and twist around each other at this point. The point where they cross each other is called a chiasma, or in plural form, chiasmata. So do take note when to use the singular and plural form when you are labeling diagrams. Also note that the strength of their coiling may cause them to break and exchange parts. This is a process called crossing over. If you have a look at the small diagram below, you'll notice that after crossing over takes place, the chromosomes for the female parent will receive some genes from the chromosomes of the male parent. This will result in new combinations of genes along the chromosomes. Finally, at the end of late prophase I, the centrioles will reach the opposite poles of the cell, the nuclear envelope and nucleolus will disappear, and the spindle fibers form. After prophase I, there is metaphase I. This is where the pairs of homologous chromosomes arrange themselves along the equatorial plane of the spindle. The two chromosomes of each pair face the opposite poles of the cell. Also note that each chromosome is now attached to one spindle fiber. If you can recall prophase I, we've mentioned about how crossing over actually helps to produce new combinations of genes along the chromosomes, right? Well, in metaphase I, we also have another process called independent assortment. This process also leads to the variation in the gametes produced. Basically, the chromosome from each pair can combine with either chromosomes of the other pair. In this diagram, we have labeled the chromosomes accordingly so that it is clearer for you. These letters represents the alleles or the genes in the chromosome. For the first combination, the small a will combine with the big B, whereas the big A will combine with the small b. So at the end of meiosis I, you will have two types of genotypes, as you can see on each side of the diagram. Now with independent assortment, the chromosomes can always switch places. In this second combination, you will notice that the chromosomes at the bottom part of the cell has actually switched places. If you're not sure, you can always pause this clip, go back to the previous slide, and compare. So, for this combination, the genotypes produced at the end of meiosis I would be different from the first combination. Thus, this is how independent assortment leads to variation in the gametes produced. Next, we have Anaphase I. This is where the homologous chromosomes separate and move to the opposite poles of the cell. Each chromosome here is still made up of two chromatids. In telophase I, a nuclear membrane forms around the chromosomes at each pole. This is followed by the division of the cytoplasm. The cytoplasm actually cleaves into two, producing two daughter cells, each with the haploid number of chromosomes. At this point, the centrioles will divide as well. We have now reached the end of meiosis I. Before we proceed with meiosis II, let's pause for a while and think about this question. At the end of meiosis I, are the two daughter cells produced genetically identical? Please write down your answers on a piece of paper. If you're not sure, not to worry, you can always pause the video clip and go back to the previous slides to figure out the answer, okay? Once you are done, let's continue with meiosis II. Similar to meiosis I, meiosis II also consists of the various phases. namely your prophase, metaphase, anaphase, and telophase. But this time round, each phase will end off with a number 2. Okay? So we'll start with Prophase II. This is where the centrioles move to the opposite poles, the nuclear envelope breaks down, and the spindle fibers appear. Moving on, we have Metaphase II. This is where the chromosomes arrange themselves along the equator of the spindle. However, if you look carefully, there is something different in Metaphase II as compared to Metaphase I. Can you figure it out? If you already know the answer to this question, well done! You can now proceed with the next part of the lesson. But if you haven't, I suggest you pause this clip, move back to the previous slides, find out the answer before you continue. This is Anaphase II. In Anaphase II, the centromeres divide. If you look at the diagram, you will see that the sister chromatids are actually separated when they are being pulled towards the opposite poles of the cell. Finally, at telophase II, the spindle fibers disappear. The nuclear envelopes form around the daughter chromosomes at each pole. The cleavage of cytoplasm will result in 4 daughter cells being produced, each with half the number of chromosomes as the parent cell. Take note that we use the term haploid to describe cells with half the number of chromosomes as the parent cells. Okay? It is now time for you to answer some questions. You are supposed to write down your answers on a piece of paper so that you can discuss them with your teacher in school. Also remember that you can always pause this video clip to refer to the previous slides whenever you are unsure, okay? For the first question, can you spot the mistake in the descriptions accompanying this diagram? Write down your explanations in your paper. Similarly, for the second question, you also need to spot the mistake in this description. Again, write down your answers on your paper. Now for the last question, you are supposed to list down the similarities and differences between mitosis and meiosis. To make things easier for you, we kind bio teachers have provided you with the diagram so that you don't have to refer to your textbook to answer this question. Congratulations! You have finally completed the online lesson on meiosis. Hope you had fun learning about this topic. If you have any more questions regarding this topic, do approach your bio teachers. We are more than happy to help you. Thank you and goodbye.